Hello everyone. We all know that genes are the basic physical and functional units of heredity. But do you know the fascinating history behind the discovery of these tiny elements that shape every living being? Today, we are going to trace the journey of gene from Gregor Mendel's early experiments with bees to Oswald Avery's groundbreaking discovery that is, DNA is the genetic material. So, let's dive into the timeline of gene manipulation. Our journey begins with Austrian monk Gregor Mendel, who in 1866 introduced the concept of the gene, although he did not call it that at the time. Mendel performed countless crosses between pea plants with different traits and carefully documented the results. From this data, he developed key principles of heredity, like the segregation of traits. But the word gene had not existed yet. Instead, Mendel referred to these factors controlling inheritance as Formbildung or form building elements. Little did he know that this work would lay the foundation for modern genetics. Next, in 1902, we meet Walter Sutton, Theodore Boveri, and Archibald Garrod. While studying chromosomes during meiosis, Sutton and Boveri noticed that chromosomes behave just like Mendel's hereditary elements and proposed the chromosome theory of inheritance, connecting Mendel's work with physical structures inside the cell. In the same year, physician Archibald Garrod provided another key piece of the puzzle. He identified a metabolic disorder called alkaptonuria and showed that it was inherited in an autosomal recessive pattern. This was the first clue that genes were not just abstract entities, they were directly involved in producing enzymes that control our body's metabolism. It was until 1909, 25 years after Mendel's death, that the word gene was introduced by the Danish botanist Wilhelm Johansen. Although Mendel had introduced the concept, the word gene was not used until 1909 when Wilhelm Johansen used term gene to describe the heritable factors responsible for transmitting traits, coining a word that has shaped biology ever since. Moving on to the year 1911, where genes became more than just a theoretical concept. American geneticist Thomas Hunt Morgan, using fruit flies, proved that genes are physically linked together on chromosomes. His work on Drosophila melanogaster demonstrated how genes are inherited together in groups, laying the groundwork for the study of genetic linkage. Fast forward to 1942, during the Second World War, George Beadle and Edward Tatum discovered that mutations in fungi caused specific biochemical defects. This led to their one gene, one enzyme hypothesis, suggesting that each gene encodes a single enzyme responsible for a specific metabolic pathway. This was a breakthrough in understanding the gene's functional role. Finally, we reach one of the most significant discoveries in genetics. In 1944, Oswald Avery and his colleagues showed that DNA, not proteins, was the genetic material responsible for carrying hereditary information. This discovery revolutionized biology and marked the dawn of molecular genetics era. In just a few decades, our understanding of genetics transformed from mysterious inheritance factors to identifying DNA as the blueprint of life. From Mendel's early experiments to Avery's groundbreaking discovery, these pioneers have paved the way for the incredible genetic advances we benefit from today. If you found this timeline interesting, don't forget to hit that like button and subscribe for more content on this amazing world of biology. Thank you.